Okay, hello everyone. It looks like we have our lectures on the Zoom call. And then thank you for everyone for Youth Neuroscience Association's summer introduction to neuroscience summer course. Over the past week, we've been talking a lot about the cellular basis of neuronal signaling, looking at the structure of cells in the nervous system, action potentials, and then synaptic transmission. Now we're going to take a more macro scale look at the structure of the brain that'll really set the stage for what we're going to be talking about for the rest of the course, trying to understand how our brain at the level of circuits coordinates complex functions such as sensation, memory, and more.
I guess the challenge, but also the beauty with neuroanatomy is that it's like a big biological jigsaw that is squashed in a tiny space, and it actually forms pathways that are extremely useful in terms of understanding how you take decisions with your brain and how those decisions are translated into movements, into actions, and how you also sense the external environment. So my background is medical, but I've come out of clinical practice in clinical medicine 20 years ago, and I joined the anatomy department in my home institution in the University of Southampton, which is right down in the south, as the name suggests, in the UK. We call it the tropical part of the UK. And there are many things that have happened in Southampton, apart from the university that opened 40 years ago. We have had the Titanic leaving here, which is not a very happy note, and several aircrafts that fought in World War II were actually invented here. So there you are, our little claims for historical fame. Now, one thing that is also happening in Southampton is we are very passionate about teaching anatomy using human postmortem specimens. So in many medical schools at the moment, the anatomy is being taught using models or plastic models or using computer programs. And it's just not quite the same. And the reason it's not the same is because you cannot get the nuances of variation. We're not all built, we're built with the same building blocks, but they're arranged slightly differently in different people. And until you actually see that arrangement in the real specimens, it's very difficult to distinguish the nuances unless you see postmortem specimens. So we're going to go through a few concepts here, but I think the first idea that I'm going to start with is the arrangement of the human brain. So what you're looking at now is a lateral view of a human brain taken out of the skull. And what you can see...
Roxana,
yes?
Roxana, your slides
yes.
are not... We can't see your slides. We can just see your first slide on Neuroanatomy, IYNA.
Ah, this is weird because I'm going through them now. So you can't see it now?
No, at least I cannot.
This is very weird. No, nothing. I'm moving through them.
Is this true for everyone?
One thing that we might be able to do,
Okay.
it's a little bit inconvenient, but if you want to send it to us, we can display them and you can just tell us when to switch screens.
So you still can't see them now?
No, we can just see your title.
Through it.
First slide.
I thought I did because I sent it to Alden, to Gabriella. I sent them already.
There it is. Okay, we can see the gross anatomy now.
You
There
can
you
see
go.
it now. Perfect.
Yes.
Okay. Excellent. Right. Okay. So you can see now the human brain, lateral view of it. And what you can see is that it has several folds separated by several depressions, if you like, between them. And these depressions are called sulci, S-U-L-C-I. And you can also see here at the back, that's the back and that's the front. So what you can see at the back is the cerebellum, which is actually one of the oldest, phylogenetically oldest part of the brain. And it is involved in balance and coordination. And right here, stuck to the cerebellum, is the brainstem. So if we were to continue down from there, the brainstem would be seen continuing with the spinal cord. Okay. What else can you see? You can see this translucent sheet of material. Can everybody see that translucent sheet of material? If I don't see a no, then I'm assuming that you can see it. I'll come back to the phylogenetic bit. I've seen that question. So this sheet, this translucent sheath, is called the arachnoid. A-R-A-C-H-O-N-O-I-D. Okay? And this arachnoid forms part of the three meninges that encompass the brain, that cover the brain. So your brain lives in the skull, and it has between the brain substance and the skull, it has this translucent bit. You see the shiny bit on the actual brain. That's called the pia mater. And you cannot lift that off the surface of the brain. It's very, very thin. It's only about 1 cell thick. And then outside the pia mater, you have this translucent arachnoid. And you can see that the arachnoid is really like a duvet, a bed sheet, because it covers the brain, but it doesn't actually go into every fold. And between the arachnoid and the pia mater are the blood vessels. Can you see the blood vessels just under the translucency of the arachnoid? Those blood vessels are therefore located in the subarachnoid space. So even though it's not a space, as you can see, it's a potential space. It's the arachnoid is closely adhering to the surface of the brain, and in between them are the blood vessels that will then dive into the brain substance. And that so-called space between the surface of the brain and this translucent matter is called the subarachnoid space. And the reason that we need to know about it is because in that space, in the living human body, there is cerebrospinal fluid, or CSF. Okay, so when you there are several major functions that the CSF has. One is as a so-called buoyancy fluid. So if you have a small head injury, as in you hit your head against a cupboard, for example, which I do quite often, then the CSF protects the brain and it doesn't allow it to become injured. However, in more severe trauma, the CSF can't cope and your brain becomes traumatized quite badly and you can get complications. I'll come to that a little bit later. So back to this term of phylogenetic before I get carried away. It means almost historical origin of the human being. Yes, how we evolved as human beings. So even animals of a very far away species from human species will have a cerebellum, okay? They won't have a brain that is a cerebrum, which is this big bit, as well developed as humans, but they will have a cerebellum because they can keep balance. In fact, there are many animals that are better at balancing compared to humans. So moving on now, what you can see on the left is again a human brain. You can see the blood vessels on it. We now know they are in the subarachnoid space. And we also know that different parts of this brain have different functions. So, for example, the front bit of the brain, which is organized as the frontal lobe, there are different lobes of the brain. This one has everything to do with personality, with intelligence, with just integrating different functions. So, for example, for you to actually understand and process and think about what you say when you speak, it's not enough to just use the speech area, because you've got to be able to understand for your speech to have meaning. That speech area connects to this frontal lobe, okay? So of course, if there are injuries in the frontal lobe, then you get major personality changes. Some people can become disinhibited. Some people can become more aggressive. So in general, the frontal lobe defines your personality, which I think is fascinating. And by the way, we don't actually know a lot of the mapping. There's a human brain mapping exercise going on with several major centers around the world, and we're still understanding the different functions of the brain. We're still understanding how they connect to each other, and we're still understanding how what goes wrong in the different diseases of the brain. So now what we do know about the mapping of the functions of the brain is that in general, the front bit of the brain is to do with motor function, which means actions, whereas the posterior part of the brain is more to do with sensory perception. So essentially being aware of the external environment. So hearing things, seeing things, feeling things, they're all more mapped out in the posterior part of the brain. By the way, do we know the difference between anterior and posterior? Anterior is front, posterior is back. Okay. Now, if you cut a section through the brain, which we call a coronal section, because it's almost like putting a crown on your head, you know, that's the plane of section. If you cut the brain in that plane, then what you see is the image on the right, okay, or at least it's the right of my screen. And you will see the first thing that I think is pretty obvious is this difference in colors. You can see sort of a grayish color, or you can see a paler color. So the grayish color is called the gray matter of the brain, and the more paler color is the white matter of the brain. So we'll try and spend a little bit of time in understanding what differences are, because this is one of the key principles of understanding neuroanatomy, the difference between gray and white matter. Okay. Now, mapping, again, going back to the area of mapping. These are just a few flavors, if you like, just a few items that we do know about in terms of the mapping of where the brain is, of where the different parts of the body are represented on your brain. So, for example, if you want to wiggle your fingers from your hand, it's from this area here that the decision is made. It's this part of the brain that will control it. If you want to make new memories, which is what you're doing at the moment, because you're listening to me and you're trying to remember things, memories are stored in a place called the hippocampus. And it's interesting because this hippocampus is hidden away in this part of the brain called the temporal lobe. Okay. This tiny part here with the green arrow is called the hypothalamus. And this one controls your body organs. So it's very interesting how these tiny areas, look, you've got a ruler here, and it shows you how tiny they are, millimeters, some of them, and they have such an important function. Now, this gray matter in the brain is mainly on the surface of the brain, but you can also find it inside in the substance of the brain. And these big areas, these big clumps of gray matter are called the basal ganglia or the corpus striatum. So no need to remember the exact, and they are formed by other structures as well. So no need to go into all of this detail. But these bits here, the basal ganglia, they control movement. And I'll come back to them a little bit later. Okay. So again, before we leave the mapping, this is again a diagram of what you've seen so far. And there are different names to the different areas, and there are different the brain is organized in the different lobes. So you've got the frontal lobe, you've got the parietal lobe here, you've got the temporal lobe here. And each of these lobes hosts different areas that mean something functionally. So for example, this Broca's area, because a man called Broca discovered it, is the area of speech. And you see how close it is to the frontal lobe, because you can speak, but it doesn't necessarily have meaning, whereas you need to have meaning with it connecting. You will only have meaning if it connects to the frontal lobe, okay? Same goes for hearing. This primary auditory cortex is essentially the area where your brain perceives the hearing. And this area here is a sensory area. So you're perceiving things, you're not doing things. So it's more towards the back of the brain, yeah? And it's very close to this other area called Wernicke's area. And this Wernicke's area is where you understand what you hear. So you can see how, if you have damage in this area, for whatever reason, you can still hear, but it doesn't mean anything, okay? Or if you have damage to the frontal lobe adjacent to Broca's area, you can still speak, but it won't make any sense to you or to anybody else, okay? So we'll come back to some of these areas. Now, a little bit about terminology, so you can actually understand better what I'm talking about as I keep going. So a synapse is how nerve cells talk to each other. It's the connections between neurons. Neurons are nerve cells. A pathway is a chain of neurons that talk to each other, and they all have the same function. So for example, the hearing pathway, or the vision pathway, or the motor pathway for movement of limbs or contraction of muscles, okay? Now, a bundle or a pathway within the central nervous system goes by these fancy names. And the only reason I've put this up is because if any of you open a neuroanatomy book to try and find out a bit more, you will see all these very confusing terms, but they all mean the same thing: a bunch of axons within the central nervous system. And if it's outside the central nervous system, it forms a nerve. So the sciatic nerve, the femoral nerve, all the nerves that you will encounter when you start studying the nervous system, they are formed by axons. They're bunches of axons, okay? Now, I know that there have been a couple of questions in the chat. So the grey matter that I showed you on the surface of the brain and the big blobs inside the brain, that is entirely formed by the bodies, the cell bodies of a neuron and the connections between the neurons. Whereas the white matter is formed by the axons from neurons. So in other words, all the heads, the controlling bodies of the nerve cells come together and they form grey matter, and then their extensions travel together as the white matter if you're inside the brain and the spinal cord. And then if they extend outside the brain and the spinal cord, which they do, they do so as nerves. So for example, you can have nerve cells whose heads, cell bodies, sit in the grey matter of the spinal cord, and the axons come together first as white matter, and then as soon as they leave the spinal cord, they can't be called white matter anymore. They are called a nerve. Okay, so you have several different nerves. If information reaches the spinal cord or the brain, it does so via sensory pathways or ascending pathways. And for information that goes away from the spinal cord or from the brain, that is forming descending or motor pathways. Okay, so there is another bit of terminology that I want to add here that I have not included in the slides, and that is the central nervous system, the CNS, is formed by the brain and the spinal cord. Brain, cerebellum, brainstem, and spinal cord. Whereas the peripheral nervous system is formed by nerves. Okay? And then within the central nervous system, the grey matter is formed by the cell bodies and synapses between the neurons, and the white matter is formed by the axons. Whereas in the peripheral nervous system, the nerves are all axons of neurons. So here we are. You've got a diagram of the CNS, the central nervous system, on the left, and you've got a little bit part of the peripheral nervous system on the right. Okay, so on the left, there's your brain, there's the cerebellum, yeah, and there's the brainstem. And what about these blue cavities here? What are they? They are called the ventricles, and this is where cerebrospinal fluid is made, is produced. It's produced inside the ventricles, so they're full of CSF, and then it escapes out here, and then it surrounds the brain and the spinal cord under the arachnoid. So this way, your brain is supported in fluid from both within and outside. Okay. Now, in terms of the peripheral nervous system, so the nerves, they are the axons. And where do the cell bodies live? They live either in here, in the grey matter of the spinal cord, or they can live outside the spinal cord in little clusters of cell bodies called ganglia. So you may have come across the word ganglion. There are different types of ganglia, but these ones here, adjacent to the spinal cord, whose axons go into the spinal cord, they are sensory ganglia. Okay, but they're different types. We won't go into any major details. So these nerves that are axons, they can be sensory, so they can carry information towards the brain and the spinal cord, or they are motor, they carry information away from the brain and spinal cord. And here's the clever thing. The clever thing is that if you're aware of the information, then that information has reached the grey matter of your brain. And that grey matter is called the cortex. So for example, everything you can see, everything you can hear, everything you can feel, temperature, pain, everything that you are aware of has reached via sensory nerves, has reached your spinal cord, has traveled up through the white matter all the way up to the surface of your cerebrum, of your brain. And that's what makes you aware of it. That's conscious. But information can also stop at lower levels, and you're not aware of it. And that is unconscious information. Doesn't need to reach the cortex, okay? And then you're not aware of it. So, for example, you're not aware of each movement that your stomach makes as it digests your food. You're not aware, and you can't control how your sweat glands function. They just respond. If it's hot, they do their thing, you sweat. If it's cold, they don't. But you can't control it voluntarily. So it's not controlled by your cortex, okay? Now, in order to study the nerve cells, you need to be able to actually view deep down inside the structure of the brain through a process called histology. So we rely a lot on histology in order to see brain cells. So here you see the blue nerve cells, neurons, but neurons only form about 25% of the brain structure. Most of the other cells are supporting cells. Astrocytes, these big red ones, they are supporting cells. And what they do is, as soon as the brain is injured, they form a scar. And these are tiny, tiny injuries. So, for example, maybe the tiniest of bleeds from a capillary, and the astrocytes will form a scar, okay? Oligodendrocytes, the green ones, what they do is they form the myelin sheets around the axons. The reason the white matter is white is because there is myelin. And myelin is nothing else other than the oligodendrocyte cell that very kindly wraps around the axons in order to make them faster at conduction. And you're going to wonder, how is this faster if they're just wrapped up? Well, the bits that are wrapped up do not conduct action potentials because they're very fatty. But these bits that are left uncovered, they do. So the way in which, if you want a fast impulse to travel down the axon from the center here, from the head of the neuron, then what you want is very fast conduction. So it comes to here, and then it jumps to these nodes. They're called nodes. And that makes it very, very fast, a lot faster than the slow tracking along the axon, okay? So remember I said to you that some actions just aren't controlled by the brain, and you're not conscious of them. And one of the things that you're not conscious of, or you can't control consciously, voluntarily, is a reflex, okay? So I don't know how many of you, you're all too young, and I don't know if any of you have had a general checkup from a general practitioner who would test your reflexes, but I'm sure most of you would have seen or heard how you can take a little hammer, and if you hammer on the tendon of your knee, then what should happen is that your leg pushes forward, jumps forward a little bit. And the reason for that is that if you hammer the tendon where it says receptor here, that tendon has little sensors called receptors. And then the information goes along the sensory axons. So the axons whose cell bodies live in the ganglion. So your cell body is here, the axon is here, it picks up information as you hit the tendon, takes it in very, very fast into the spinal cord. It synapses here, there's a lot of activity here in the gray matter, and then immediately another neuron sitting here will send the information out through the axon towards your muscle, towards your quadriceps muscle that contracts, and therefore you kick your leg. If anything along this pathway is damaged, then this reflex won't work. So it's always, always tested after accidents. You know, if there's been a road traffic accident and the patients are in accident and emergency room, then their reflexes will definitely be tested, because you want to know if any of these nerves have been damaged. Okay? So this, of course, this is, I think, a better diagram. It's more complete, where you know, you can also see a very similar reflex, but instead of it being the lower limb, it's the upper limb. So what happens, I wish you could talk to me, but of course you can't. What happens if you accidentally burn your finger? So I cook dinner, and very often I'm in a rush and I'll put my hand in the oven and I may burn the bit of my forearm that isn't covered by a glove. And then what happens is, of course, I withdraw my hand very quickly, because, and the reason for, I don't even think about it. Oh, I'm burning, I must move my hand. It's reflex, it's involuntary. Yeah. Eventually I'm aware I've burned my hand, but you actually move it faster than you're aware of it. So your receptors, your sensors in the skin will send information very fast through these blue axons that belong to the neurons sat here in the ganglion into the gray matter of the spinal cord, and then immediately the information goes out to the muscle to withdraw your hand. Now, there are a number of conditions. I'm keeping my eye on the chat. There are a number of conditions where something can go wrong in this pathway. So you can have damage to the nerves because of trauma, or you can have different conditions, for example, multiple sclerosis, where axons are damaged because the myelin is damaged. So this whole thing is slower. So there are lots of different conditions. Or if the blood supply is cut off by a stroke, then none of this works, because all of this takes a lot of energy and you're relying on blood supply on efficient blood supply for it to work. Just like you have nerves going in and out of the spinal cord, you have nerves going in and out of the brain. To be more specific, the brain stem. And so the brain stem is a small area, it's a very busy area. It's got both gray matter and white matter. And cranial nerves, there are 12 pairs of cranial nerves, go in and out. So you can have the optic nerve that serves vision. You can have oculomotor nerve that serves the muscles of the eye, so you can move the eye. Cranial nerve 3. You've got nerves that pick up the sensory information from your face, from your tongue. That's sort of hot and cold general information, but you can also have taste, which is specialized information from your tongue. That's a different set of cranial nerves. You can have cranial nerves to do with swallowing, with supplying the muscles of your pharynx. You can have cranial nerves that are, so another function of a cranial nerve is to do with speech, so to move the muscles of your larynx. And one of the busiest cranial nerves that does lots and lots and lots of different functions is cranial nerve 10, which is also called the vagus nerve. The reason that's very busy is because it goes all the way down to your lungs. It supplies the bronchi. It goes all the way down to your stomach. So it supplies the muscles of the stomach and it helps with digestion. So it's a very long nerve and a very busy nerve. Now I started with the skull, and I'm coming back to the skull. This is the base of the skull, and it's actually one of our skulls in where I work. And you'll see that the skull has different holes, and these holes are for these cranial nerves to go in and out, because obviously they need to be able to reach the brainstem. Okay, and you see they're connecting to the brainstem. Okay, now back to the mapping. I showed you right at the beginning that one of the long strips of tissue, which is called a gyrus. The gyri are these strips of tissue, and the sulci are the depressions between the gyri. So you've got gyri, which is the plural for gyrus, and you've got sulci, which is the plural for sulcus. So one of the long ones is this post-central gyrus, which is just, the reason it's called post-central is it's just behind the central sulcus. And on this strip of brain, your entire body is represented. So it's a sensory representation. So what you feel from the different parts of the body is represented here, but it's not represented in a similar manner to how big that area is. It's represented according to how sensitive it is. So if you think about what you can feel with your hand compared to what you can feel with your foot, the area for your hand is much bigger. The area for your face, your tongue, that's why he's got his tongue stuck out. It's because you've got lots and lots of neurons that pick up information from those parts of the bodies. So your hand and your face and your tongue and your larynx are all very well represented. They occupy a lot of space, whereas even though your thigh is bigger than your face, it's not actually that sensitive. If you put two pins close to each other on your thigh, you can't distinguish their two. You'll feel they're more like one, whereas you can on your face. So I've shown you how you can cut the brain coronally. This is cutting it if you were to go with a saw through the nose, separating the right and the left nostrils. That's called a sagittal section. So here you've got a sagittal section through your brain. And again, you can see the cerebellum, you can see the gyri, you can see the sulci. You can see the white matter that connects the left side to the right side of your brain. And you can see the brainstem. You can even see some of the ventricles here, where the CSF is produced, and a little bit of the arachnoid. Okay? And remember that each and every little bit of this brain has a different function. So I know that this is neuroanatomy and not neuroembryology, but it's really important to understand how the brain is formed in order to work out how it's built the way it's built. So I'm not going to go through any details, but I will just say to you that at 20 to 21 days after the embryo is formed, when most ladies don't even know they're pregnant, 21 days, they haven't yet tested, they don't know that they've missed a period, that's when the nervous system starts forming. And this is when most of the problems, most of the neurological defects, most of the abnormalities happen because this process goes wrong, and I'll show you. And a lot of it is because people don't even know that they're pregnant. So an embryo has 3 layers, this red layer, green and yellow that you can see here. And the top bit, this yellow layer, starts folding, okay? So it folds and folds and folds until eventually the margins of the folds come together. And what is supposed to happen is that they are supposed to close and form a tube, something called a neural tube, okay? And once it's closed, then different parts, see there's your tube, and then you've got the different grey matter and white matter surrounding forming. And note that in the spinal cord, the grey matter is in the middle and the white matter is on the periphery, whereas in the brain, it's the other way around, okay? Ignore the specific terminology. And then another thing that's supposed to happen is this tube is supposed to close at the front, okay? And then these bits here are supposed to thicken a lot, fold so that they can fit into the developing skull, yeah? And here we are, an embryo at 8 weeks, 2 months. Already it has the beginnings of the central nervous system. But because it started as a tube, it kind of still stays as a tube, and that's how the ventricles remain with that fluid inside, with the CSF inside. And then eventually these things start folding and developing more and more and more until you get the gyri and the sulci. But sadly, sometimes you can get conditions like you can see on the right, which are incompatible with real life. So here, the brain didn't form at all, okay? It just did not close at all at the front of the neural tube. And the condition is called anencephaly. So the fetus is just aborted. It just comes out dead. Other conditions are where the neural tubes have either closed, like it's closed here, but the problem is that the bone around it in the spinal column hasn't closed, and that's called spina bifida. And you can live perfectly normally. In fact, you don't even know you've got, most people don't even know they have spina bifida occulta, because there's nothing wrong other than the fact that the bone here from your spinal column hasn't closed, okay? But you can have variations of this, more and more severe, where the bones haven't closed, but then this bit pops out of the neural tube, or maybe the whole spinal cord pops out, or it just hasn't formed at all, it hasn't closed. See, the open neural groove means that the neural tube hasn't closed, which is what you can see on the right. And again, this is incompatible with life. Okay, so we went a little bit about how the brain perceives the different parts of the body. What you see here is a diagram about how the different nerves, here you've got a cranial nerve, here you've got a spinal nerve, another spinal nerve, they pick up information, go up through the white matter. In the sensory pathways, information just cannot reach the cortex directly. It always has to relay in a place called the thalamus. So the thalamus is the relay center of the brain. Any information that reaches the surface of the brain has to first stop through the thalamus. The motor pathways are exactly the same, but the other way around. So this is how your brain perceives your body that it controls. You've got a better diagram here. And funnily enough, it looks a bit like the sensory gyrus. The only difference is that this is called precentral gyrus. That means it's just before the central sulcus. And again, most of the fine muscles of your body, where you can perform fine movements, this is not about how big your muscle is. It's about how refined the movement is. So obviously, muscles of the face, the tongue, speech, hand, yes, they're all very sophisticated. They require a lot of input from the brain to function, and they occupy a larger area on the brain. Okay? So again, here you can see traffic the other way around. You can see how a decision is taken up here, and then information goes down to either the brainstem or to the spinal cord, and then from there it goes out through a nerve. One thing that I want you to notice is that as the information goes down into the spinal cord, it crosses over to the other side. So the consequence of this is that if you have a stroke on the left side of the brain, it will be the right side of the body that will not be able to perform movements. Okay? Now, all of this, sensory, motor, etc., this requires a huge amount of energy. And this energy has to come from blood supply. So because the brain uses so much energy and oxygen, it receives 1/5 of the blood from the heart compared to the rest of the body, even though it's very light compared to the rest of the body. And it consumes a lot of oxygen. So in other words, it can't live for long. The brain substance starts dying if it's deprived of oxygen for long. Okay? So the two main suppliers, here what you can see is the brain upside down, is a human brain viewed upside down. You can see some stringy bits, which are the nerves going in and out of the brainstem. But you can also see these translucent tubes, and they are the blood vessels that come from the neck and go into the brain. And there are two main suppliers of blood. One is from the carotid artery, the internal carotid artery, and the other one is from the vertebral artery. These two main arteries then start dividing into these arteries that are in the subarachnoid space. Remember, in between, sandwiched between the arachnoid and pia mater. And then they dive into the substance of the brain, and then they start dividing, and they divide into smaller arteries called arterioles, and then they eventually divide into capillaries. And the most important aspect of the arteries of the brain and the capillaries of the brain, actually it's the capillaries rather than arteries, is this concept of a blood-brain barrier. So the brain is equipped, remember the good old astrocytes, it's equipped with these astrocytes that stick their feet, they actually are called astrocyte feet, onto the walls of the capillaries to prevent anything toxic coming out into the brain. The problem is that things like carbon dioxide get through, things like alcohol get through, things like drugs get through, but useful things such as chemotherapy for brain tumors, they don't get through, okay, because of this blood-brain barrier. And this is a major challenge for treatment of brain tumors, because you just cannot get the chemotherapy in there. So the wall of the blood vessel is a very busy place. It has the lining, the endothelium, obviously that forms the lumen through which blood goes through. It's got little contractile cells that make it pump, called pericytes. It's got these astrocyte end feet that hold it together as a blood-brain barrier. And the wall of an artery is even busier because it has several layers in that wall. Okay? But it also needs to be, so it needs to be contractile, it has a lot of muscle, and it needs to be elastic, so it has elastic fibers. Now, we're done with the actual concepts of neuroanatomy, and what I'm about to show you are some pictures from the pathology department. So what happens when things go wrong? What you see here is the same patient. This is a lady who was in her 30s and she fell off a horse. And she didn't wear a helmet. So, and the same thing we've seen with motorbike accidents and bicycle accidents. So the skull is cracked. There's a fracture there. Because there's a fracture there, then the blood vessel that is in the subarachnoid space, that one started bleeding. You see all this blood here. That blood has started to coagulate and form this big hematoma, and then the brain itself has been damaged with all these big hemorrhages in the brain. So that's what killed the patient. Okay. Now, you don't necessarily have to have that major bleeding. You can have the brain sort of hitting the skull and then bouncing back and hitting it again. And that action stretches these axons and it damages them. It tears them completely, as you can see here. Okay? The other thing that can happen is that these nerve cells can start dying, and the normal proteins that are in the brain can start building up as toxins, toxic plaques in the brain. This has been discovered first by Alzheimer in 1907, and eventually that leads to dementia, which is where you're starting to lose the normal cognitive functions. So there are many types of dementia, but the most frequent type of dementia is Alzheimer's disease. And what you can see on the brain is that the brain is shrinking, the brain matter is shrinking. These gyri are smaller and more wider apart. The sulci are bigger. Okay, so there's a coronal section through an Alzheimer's brain, and there's a coronal section through a normal brain. So you see it's very stringy here, and there's a loss of brain substance. Then when you look inside the brain by histology, you see these buildup of very toxic proteins that just start killing the nerve cells, forming tangles or just big sticky amyloid plaques. And even worse, they build up in the walls of the blood vessels. So they don't allow the blood vessels to perform their function normally. So how do we investigate neuroanatomy and, histology? We take sections through the brain, through the post-mortem brain, and we look at them through confocal microscopy or electron microscopy. So with the confocal microscopy, you can see fancy things like blood vessels on the surface of the brain, and all the red is the sticky amyloid that shouldn't be there. The green is the smooth muscle in the wall of the artery. That one big thing is an artery here. They're the muscle cells in green, and the red is the toxin that is building up. Okay. Now I'm a clinical neuroanatomist, so I deal with dead brains, post-mortem brains, either looking at them macroscopically, the whole brain, or microscopically, like you can see here. But fortunately, we have people like Dr. Agarwal who actually can investigate the brain with fancy new techniques that didn't even exist when I was in medical school, in the specialty that is now recognized as an independent specialty called neuroradiology. So I just wonder if right now the best thing would be to hand over to Nivedita Agarwal and then take questions at the end, or do you want questions for me? And then I'm just aware that she might be busy. Niv, what do you think?
So, ma'am, it was an excellent talk, and we really enjoyed it. So, I was wondering if a question session would be good right now, and then for you, the questions that are there, if any students has any question right now for you, and then then we can have questions for Dr. Nivedita as well.
Yeah, let me just check that Dr. Nivedita actually can speak in a few minutes.
Sure.
Because I'm, yeah?
Yes, I am here. Hello, everyone.
So,
I think my suggestion would be that I go through my slides, and probably I'll be able to answer some of the questions you might have right now. After that, you could have, the questions compiled at the end for both. I suspect some of the questions may be answered by me during the talk.
Yeah. So some of the questions that you might have might actually be answered in Dr. Agarwal's talk. So be a bit more patient and hold on to them, and let's see what we mop up in terms of questions right at the end. Yeah?
Sure, ma'am. Sure. That would be a good idea. Okay.
I'll mute myself now.
All right. Let me share my screen.
And I'll stop sharing my screen so that, stop share, I should be out. So you can't see my slides anymore, I hope. Ah, perfect. Right, I'm going to mute myself.
All right. Okay, well, hello everyone. Thank you for the invitation, Roxana. Let me give you a brief overview of who I am. So I'm a neuroradiologist working in the northeast of Italy. I was asked to do this talk an hour before I was going away for my long weekend. And I agreed to do this talk for 3 reasons. Number one, my respect and great friendship with Roxana Carrare. I just can't say no to her. Number two, because I am passionate about teaching. I have given several lectures to students at all levels of their academic careers. I had a great biology teacher when I did my A-levels 20 years ago. And the reason I scored an A+ was mostly because of him. And I vowed to myself, if I ever give a lecture, it should be good enough so that they remember, it would make a difference in their careers. And number three, this is my first ever chance to talk to UK students, which is great. So one more thing, I actually was sent the slides by Roxana, again, an hour before I was leaving. I actually never looked at them. I just went through them right now as she spoke, and I'm amazed how similar our talks are. You know, they're complementary, they're not similar, which just means that we think alike. All right, let me see if these are going to work. They're going to be just 4 to 5 slides on neuroembryology, and I'm grateful to Roxana that she talked about this, because I think any study on neuroanatomy cannot be done without having a very, at least a brief understanding of how the central nervous system develops, because it's a 3D structure, right? I mean, it's in the sense that it has several folds, and it can be not very intuitive to understand where what's the dorsal part, what's the ventral part, what's interior, what's posterior. So we know there's a disc, there's an embryonic disc. And what happens if you look at it lengthwise, it lengthens, all right? So you have an anterior portion and you have a posterior end. What happens is there's a layer of cells that will fold over, all right? They will fold over, and as it folds, there's a tube that forms which detaches itself from the other layers. This neural tube is where the central nervous system actually develops. So here it is. Pictures actually can reveal a lot of understanding of how this happens. So you can see that there's an anterior portion, anterior neuropore, and there is a posterior position. And what happens over the days is that it's just kind of zips. It closes like a zipper, and it closes in the anterior end, and it closes in the posterior end. Any damage to these two portions would either cause an encephaly, you saw that in Professor Carrare's slides, or you could have several levels of severity of spina bifida in the spinal cord. Now, this is your 3D folding. So again, this is your anterior neuropore, and this is your posterior neuropore. What happens is that the anterior neuropore, it folds over such that what you see on the top of your head, the surface of the brain, let's say, this is your dorsal aspect, and what folds in and actually is positioned on the skull base is your ventral surface, all right? And this, of course, is your spinal cord. So I'm, again, hoping that you get some more terminology right. So you have dorsal area and there's a ventral surface. Your back is your dorsal surface. Your inside is the ventral portion, even in the spinal cord. More images, again, to show you the spinal cord, the neural tube unrolled. What you're seeing in the top portion here in yellow and green is the cerebrum, the cerebral hemispheres. And as it folds, you will see the eyes, they come out in the front and they're not on the side, okay? And then you have the brainstem. You've seen this again in Roxana's slides, and which continues as a spinal cord. This is a coronal MR image, and we will get to this a little more. Excuse me for some of my Italian terminology. I think this is the only slide where I missed translating them. And once again, you will see your cerebral hemispheres that have the frontal lobes, that have your occipital lobes, parietal lobes, and the temporal lobes. This is the topmost part. Most of it is dorsal. and that is your thinking cap. Again, you can see the folds, several of them, several flexures. So you can imagine how complex the system is. And as this folds, what is also happening is that there are cellular, if you section this tube, let me go one slide back. If you section this tube longitudinally, what you will see inside are lots of little cellular proliferation, cellular masses that proliferate. And as they proliferate, there are axons that are being formed. And as they form and grow, they will connect to other axons and therefore to other areas. This slide here, this image on the right that you see is actually an MR image of a fetal brain. And this is at 25 weeks. So this is 25 weeks. This is 23 weeks. And you can see the cerebellum that has already been formed. You can see the brainstem. Brainstem is formed of 3 little parts. Doesn't matter right now. We just need to be as simple as possible. So this is your brainstem. This is the frontal lobe, parietal lobe, and the occipital lobe right there. The dark area is the ventricle. Again, this is a fetal brain, so it's out of the skull. And it's the MR anatomy. And as I said earlier, you'll have different cellular masses that they start to proliferate, axons start to grow, and they start to connect to each other as the neural tube and as the fetus starts to grow. Now, how do we study the brain? There are different ways of studying the brain. You could have blood samples and look at all kinds of molecules. You can do a CSF tap. You can actually remove some liquid from the spinal portion in the CSF space around the spinal cord and test for different molecules again. You could, as Professor Carrare does, look for differences in the histological specimens under electron microscope, confocal microscope, etc. The problem with that is that the person is already dead. It's a post-mortem analysis, right? But that's a very good way of learning and looking at brain tissue. You can do all kinds of fancy other tricks we do in the clinical in the clinical world, electroencephalogram, etc. We do have a lot of animal models that can mimic human diseases, and therefore we can study what actually might be happening in the human brain. But remember, some of the pathologies cannot be actually reproduced in animals. For example, suicidal thoughts. Right? How do you do that? So there are certain conditions that absolutely need to be studied in the human brain. Well, last but not the least, you can always crack open the brain and see what's going on. Or probably a less invasive technique would be to do an MRI. So this is the MR scanner that we have in our hospital. It's a very simple, very powerful though, magnet, which is 1.5 Tesla in strength. This is all terminology that as you read and get curious about, you kind of relate to this. So this is a 1.5 Tesla scanner, big, huge magnet. The patient is positioned supine inside, and then we start to capture images. What we use is these are your electromagnetic waves. They're very safe with respect to nuclear or even ionizing radiation you could use on a CT or an X-ray machine. So they're very safe. This is the first image ever of an MR in 1982. So really the technology is old, 40 years, which is not a lot of time, even though in the last 20 years, the progress in MRI has been tremendous. So this is an image in 1980s, as you can see, and these are kind of images that you can actually see today. You can already start to look at a difference in the signal intensity. That is what we say, signal intensity between the gray matter, which you can see is a little more white than the white matter, which here is represented a little gray. You might feel confused why the white matter is not more white than the gray matter, but this is just MR technology that we will not go into any further today because that would be an MR class, right? What I want to show you is the detail that MRI can show us in live human beings between the gray matter and the white matter, the ventricles. We'll go into the anatomy a little more deep, and you can also see lesions in this patient. So anatomy, this is a coronal image. I hope you can navigate this. I apologize. I mean, it's always nice to be in the class and, you know, feel the energy, which I miss while I talk to this computer. So again, what you can see here is you can see the skull, which is really dark. It's right here. The white is the skin, all right, the scalp. This is the dark portion is the skull, and just underneath the skull is a white layer that you can see here, which kind of fills these sulci or sulci. That is your water or the fluid, which is the cerebrospinal fluid in the subarachnoid space. This is all terminology that Professor Curare went through in her slides. You can again see the gray matter, just the outside cortex, and you can see the white matter, which is filling the inside. The white matter is made up of axons and all those little cells like the oligodendrocytes, like the astrocytes. That is what is constituting white matter. But the majority of it is myelin, which is fatty material around the axons. And the gray matter are cellular bodies, the little cells, and that is where the action potentials, they actually begin. In the depths of the brain are these little white cavities. Those are your ventricles. Again, that is where your CSF, the majority of CSF is produced, and it goes down and out again to the subarachnoid space and down in the spinal canal. This is your brainstem, and just behind is the cerebellum. So again, different ways of looking in using the MR to look at different parts of the brain. This extreme image is a CT scan. And probably you will realize that a CT scan doesn't have that level of resolution that an MR has. And there are disadvantages and advantages of both. This can be, we can discuss it in the Q&A, or again, this is more of an MR lecture. So right now, just remember that CT, you can do as computer tomography, which would take 5 minutes to scan the entire brain, and MR uses about 15 to 30 minutes. So it's a longer imaging technique. You can see, again, white matter, gray matter. You can see vessels. This is your aorta, and these are vessels coming off the aorta. So you can see, again, the level of resolution in just doing an MR. This is your sagittal brain, okay? This is a sagittal scan, and you can see the frontal lobes, you can see the parietal lobes, and the occipital lobe, which is right here. Look at the cerebellum, the brainstem, as it continues down into the spinal cord. This white matter huge bundle, this is the biggest white matter tract in the brain, is the corpus callosum. Corpus callosum connects the two hemispheres, left hemisphere to the right hemisphere to left hemisphere. And there are a lot of other new techniques that are now available where you can not just look at the morphology of the brain, but also the function and the function of the brain parts, and also look at the fiber tracts, which is very interesting. And I will show a little bit more detail on the fiber tracts. All right, so this is unfortunately not working, the video, which is too bad. All right, so again, just images to give you an idea of how MRI can actually look into several aspects of brain function. So the other thing that you can do, you can see is, for example, the volume of the blood in a particular region, which could be high or low depending on the pathology. Helps me make a differential diagnosis, whether this is tumor because there is a lot of blood, or it's a stroke, which will not have a lot of blood. You can also look into the chemical composition of a lesion, of what is it made up of. Is it made up of a lot of lactate? Is there a lot of alanine? Is there a lot of choline? These are all products that vary in different lesions. So what is the difference very quickly between CT and MR? So CT is the image on your left, MR is the image on the right, and you can see that this is the same patient, same size, and you can see the level of definition that you have with MR than on CT. CT, you see these dark little dots here, which is the loss of tissue, and here you can see them as really bright lesions, but there are several others that you might miss on a CT. This is a CT of a patient with an ischemic stroke. You can pretty much, it's very difficult to see it. In the initial stages, it may actually be negative. This is hemorrhage, lot of blood in the brain parenchyma of this person. Again, cerebral vessels, you can see a fine anatomy. This is not the resolution that you could see on histology, of course, but it's a very good resolution. You can see the vertebral arteries right down here. You can see carotid arteries, middle cerebral arteries, all these arteries that again wrap around the cerebral surface before the branches dive within the brain parenchyma. So MR of a stroke. You look at these images and you might not capture any lesion. I mean, this pretty much looks symmetric. There doesn't seem to be any big difference. But if you do some fun images, fun techniques, you might actually see that there is an area of the brain that actually is infarcted, right? So there's a lot of progress that is being done in MRI as I speak, really. Which has made it more easier for us to diagnose pathology even at an early stage. This can be positive in about 15 minutes of a stroke. MR gives a whole picture. So this is what we call a diffusion weighted image, right? This is an area, everybody can see this. This is the area of stroke. But this patient also has a lot of other little lesions. Okay, these are the scars. These are the astrocyte gliosis. Okay, this is when you have little damage, and what happens is astrocytes kick in and they start to proliferate, forming a scar. And these are scars in the white matter, which will disrupt with age. This will disrupt connections and therefore fluidity of thought, can have all kinds of cognitive dysfunction. I apologize, but videos are not working. This was done on a Mac, which is too bad. Oh, this is working. So, all right. This was an entire sagittal brain. This is not going to work again. Let's see. Yeah, sorry about that. All right, this is a axial MR of a normal, this is the left side is normal, and on the right is Alzheimer's brain. What are we seeing? What is the first thing that comes to our mind? That in the Alzheimer's brain, a lot more of atrophy. You can see this is a Sylvian cistern. All right, it's right here. It separates the frontal part from the temporal part. These are actually enlarged. That is basically because there is atrophy happening within the brain with respect to a normal brain where you can see the cisterns to be very nice and tight. Even the ventricles seem to be enlarged. And that is because in proportion, you are going down with the gray matter and the white matter so that the CSF spaces are enlarging. So this is how we kind of understand that there is a neurodegenerative process happening. This is true for Alzheimer's disease, but there are other degenerative diseases that can do the same thing. This is it. This again, on the left you have a normal T2 or normal coronal image, and on the right you can see a brain of a patient with herpes encephalitis. And what you see is the damage of the temporal lobe here. All right, the frontal lobes are up here, you have the temporal lobes here, the cistern is here, the Sylvian cistern, this is the temporal lobe, and you may be able to see the hippocampus here. This is the hippocampus, all right? This is a patient with major car accident. What you're seeing here is important frontal lobe damage. So all of this dark spaces, this should have been brain parenchyma, but now it is just CSF filled because there is loss of tissue. And this is bilateral, bifrontal damage in this patient. And of course, he had all kinds of cognitive dysfunctions, as you heard from Professor Carrare, social disinhibition, aggressiveness, etc. I did not prepare slides for the peripheral nervous system or the spinal cord. I just have this slide, which I prepared while Roxana was speaking, where you can see that you can image the peripheral nerves, or these are called peripheral roots in the spinal cord. This is a patient who had a meningocele. It doesn't matter. It's an accumulation of CSF, an abnormal accumulation of CSF. And you can see the spinal cord, and you can see these little dark linear objects. These are your nerve roots that come out of the spinal cord and then go out into the skin or the muscles. White matters, right? When we have been taught that intelligence is people who have a lot of gray matter. Now I can assure you that white is important. White is very important. This is an image I found which I thought was interesting for my UK students. So what you're seeing in this is that different areas of the brain are like little stations, and they're all interconnected. Okay? These interconnections are absolutely important for information to start and end in different areas. They need to be talking to each other. And the fibers, they go front to back to front. They go laterally, right to left, left, right, and then they go cranial caudal, so top, bottom, bottom, down, ascending, descending pathways, etc. So this kind of information, the connectivity of the brain, the structural connectivity of the brain, is something that we can see in the brain with the MRI as well. Quick recap of what Roxana talked about, the motor homunculus, right? It is not how much muscle you have. It is how important that muscle is that is represented here in a coronal size. So the fine movement of your hands is very well represented with a large mass of tissue, a large part of the brain, and this is lateral. Right? The more lateral is the face, then you have the hand, and in the middle part you have the lower limbs. Why am I showing you this? This is, okay, this is diffusion tensor imaging. I apologize the video is not working, but as you probably saw, okay, just one of the slides just to show you that you might be aware of novels by Oliver Sacks, and they are much to do with disconnectivity. So disconnection syndromes can give all kinds of very curious symptoms. So going back to the motor homunculus, this is a patient, an 80-year-old lady who had an acute stroke, and you might now be able to see the stroke, the really bright area that is damaged in this patient. Now, what do we think this patient may have? Lower limb, hand, face. So what we can do, so this is your homunculus again. See, the lateral side is face and hand, and then the legs and the lower limb is in the middle. If you construct fibers using MRI information, we can see that the lower limb are well connected to the spinal cord, so there is no problem in the movement. What is damaged here is the connection between the hand area and the spinal cord. All right, so this is the beauty and the power of MR, even in clinical situations. I'm actually making this a little simple because not in every case we are doing this, or we do this at a global level, but definitely this has given us the information and confirmation in our patients that this is how the brain is connected. This is a DTI, diffusion tensor imaging of a brain in a patient after rehabilitation. He had a stroke on the left side, and you can see some of the fibers now reconstructed. More DTI images, again, as you will see, the red area here is the corpus callosum. Remember that big bundle that connects the right and the left hemispheres, and bilaterally, of course. This is your corpus callosum right here. These are your axial images. This is a patient with Parkinson's disease, but look how beautiful the fibers look. Believe me when I say this, because you may not know what normal is, but this is a nice, healthy, plump fiber tracts. So the green are the fibers that go anterior to posterior and back. Red go laterally, and the blue ones are the ones that connect cranial caudal, so the spinal cord, to the brain, and vice versa, your ascending and descending pathway. Look at the Alzheimer's disease patient. Again, you will see the atrophy and the thinness of of these fibers, and the corpus callosum actually tells you all the story. Going a little more fast so that I have time for questions. These are, again, pathologic images. This is a child who had hypoxic damage at birth, and he had problems in developing most of the fibers. You can see how thin the corpus callosum is. And this is our patient with the major car accident who basically doesn't have any connections from the frontal lobe back to his occipital and parietal areas. So these were videos which are absolutely not working, unfortunately, but see how the chronic comatose patient is. This is a patient who has been in coma for about 2 years now. Well, here it is, so it is working. And you can see how many of these connections, they actually die over time. And this is absolutely important, and MRI can show you these things. Okay. This is just a curious way of showing you how this is a 41-year-old lady with this huge lesion. And you can see how the fibers are just pushed away. All right, this is just a lot of water. They're just pushed away. In fact, in just about 3 months, there was an important recovery of all the fibers as well. So these are kind of things that images can help. This is a case of tumor. You see these lesions here, and DTI can actually help understand what is happening to the fiber tracts or bundles of nerves or axons in the brain. All right. The last part is functional anatomy. So we saw the morphology in scales of gray. We've seen the DTI in RGB in colors, where you can see the direction and the integrity of fibers with using diffusion tensor imaging in MR. You can also kind of look at the function of MR, especially the major functions. Where is your language area? Where's your hand area? If there, you can also do fun things like, how is memory responding? How do you react to faces, sad faces, happy faces, etc. Let me just give you a very quick overview of what it means. Now, this is an image that's coming off internet, of course. No, it's not neutron, it's neuron, I'm sorry. One neuron would be one function and it would be great, right? That's not true. We just have, how many neurons we have? I think we have about 70 billion neurons, so that's a lot. So the concept of function in the brain is not new to us, of course. It started kind of, this science was termed phrenology, and the Americans came out with this, that areas of the brain, there were different areas of the brain, this is 1848, areas of the brain that did specific things. So for example, here you would see that all these little areas, so this is veneration, benevolence, there's another one for ideality, cautiousness, friendship, destructiveness. Of course, this is obsolete. This is not true. I bring this to you because this is how, this is the first way of, really understanding of giving function to single bits of the brain. This is not true at all. Now we have a different functional mapping of the human brain. The way we do fMRI is based on this little hypothesis. If you lie on a seesaw, right, if you start thinking, the idea is that there would be an increased blood volume in the brain, which would tilt the seesaw on the other side. So if the patient is on a seesaw, balanced, just lying down, and we ask him to start thinking or start to do something, this is important. If he starts to do something, there will be an increased blood flow within the brain, which would shift the weight towards the brain, and there will be, the seesaw will tilt, right? This is what is understood as happening today. In this patient, this is a 30-year-old student of mine who gently did what I told him to do, and this is what he did. I asked him to be in the MR scanner and just finger tap. If you can just look at the video, of course. So this is what he did. So it was just finger tapping without moving any other part of the brain. So if he did the right or the left hand, right? So if he did the right hand, then he would have these are the motor areas that are being activated. If he uses the right hand, the contralateral brain hemisphere, that starts to get increased blood. It needs a lot of blood because those areas are working, they need oxygen, right? So those, they just light up. There's increased volume, and in MRI, I can pick this up. So what you're seeing here is these three areas are the motor areas. So this means that the hand area, the left and right hands, fingers, the movement, the muscles of the hand, they're actually located in this sulcus right here. So this is the left hand, this is right hemisphere, this is left hemisphere, this is the left hand, this is the right hand. And watch, there is this third spot in the middle, which is connecting the areas, both right and left, and this is in the corpus callosum. So this is the kind of experiments that have been done in the past 30 years or so. So 1990 was the first experiment. And now we have a global understanding of where language is, where our memory is mapped to, how we react to emotions and things like that. This is our patient who had the acute ischemic stroke, remember? So if I ask this patient to move just the non-damaged hand function, and there it is. We had the nice little blob. There was an activation of the frontal lobe, the contralateral frontal lobe. But when she tried to move the paralyzed hand, I told her to try as much as she could. There was absolutely no activation. So these are the kind of things that we can do for troubleshooting, differential diagnosis. Interesting aspects, especially in cognitive science, is neuroplasticity. When there's a damage, there are all kinds of rehabilitation techniques. I'm talking motor, but this also works for cognitive dysfunction. This is a technique where you tie the normal hand and you force the paralyzed hand to operate. And this is what happened in weeks. So you have activation of the contralateral side. You can also have brain-machine interface. So you have the machine or the computer to help you activate the paralyzed muscles. And then you can either with electroencephalogram or also fMRI see if those neurons are back and functioning again. You can do this in patients who are unconscious, and you have to probe into their minds because they're just unconscious, as we have no idea if they are actually listening to us, but not able to respond. So we can kind of do all kinds of paradigms. We can ask them to imagine playing tennis or imagine navigating in their house and see if their brains actually light up. And sure they do. Patients who do respond, whose brains respond, they actually come out of their coma after a few years. This is pretty much it, and I am happy to take questions. I did put this just last minute. It's an atlas. It actually is meant for medical students. This might be too much for you, but if you are interested in anatomy, there are lots of, there's a lot on the internet, but beware to go through books and articles that actually are scientific so that you don't get dissuaded. So thank you very much.
Thank you, ma'am. That was an excellent talk, and I'm sure everybody enjoyed this. And so we'll have the questions now. And those who have any questions, please raise their hand, and then we can call out their names, and thereby they can unmute themselves, and if convenient, they can also turn on their video so that we can have. Yeah. So Ibrahim comes first. Can you unmute yourself, and if it is convenient, you can turn on your video as well.
Hi, so I wanted to ask, what's the difference in MR of a right-handed person versus a left-handed person?
I'm sorry, can you repeat that again? What is the difference between?
What is the difference between in the MR of a person who is right-handed versus as of those who is a left-handed person?
Yeah, that's a good question. So there are people who just, they are left-handed or right-handed. That's true. I'm not sure exactly the percentage, but it should be somewhere between 90% of left-handers. They actually are exactly like the right-handers in terms of their MR connectivity. So even so for example, language is, the dominant hemisphere for language is the left hemisphere. This is for right-handers. This is true for left-handers as well. But we cannot take this for granted. So for example, if they go into surgery, they will do an fMRI and we will check for the dominant hemisphere in these patients as well. But usually should be around 90%. That is how high it is that left-handers are actually wired similarly to right-handers. Suman, we can't hear you.
Thank you, ma'am. So is there any other question? Also, Professor Roxana is here. And so is there any question that we have for her?
Don't be shy.
By the way, I'm sure that, I mean, everybody in the chat box is thanking you and Professor Roxana. So
Yes,
for them,
I
I'll give you a clap. I'll give.
thank you.
Yeah,
Thank you very much.
That was really a very, very nice talk. And
Oh, we've got a question. Can an accumulation of grey matter lead to high intelligence? I wish, because I'd accumulate grey matter. You know, I'd start shopping. No, nobody really knows what the definition, the anatomical substrate of intelligence is, but what we do know for sure is that it depends on the number of connections. So, you know, the more, you know, you're intelligent through your ability to accumulate information, quickly analyze it, and take the right decisions. And all of that happens with all the connections that are happening all the time with our brains. So keep your brain active, then you'll be
Yes,
more and more.
Completely agree. The more strategies you build during lifetime to do the same thing, the better it is, because that is how you increase the connectivity. That is the entire concept of neuroplasticity. You know, we are born with so many neurons, and the moment we are born, we start to lose them as well. What grows are the connections, and yes, some of the cells as well, but really it is the ability to navigate in space, in all kinds of atmospheres and places, and is what makes a healthy, intelligent brain.
I have a question.
Yes.
Hello.
Yeah, so what will happen if the supercolliculi of midbrain is damaged?
Superior colliculus.
Yeah.
Roxana, do you want to take this or should I?
Yes, I'll take it. So the colliculi of the midbrain are the sites where reflexes, some of the brainstem reflexes are closed. So for example, the superior colliculi are for the reflexes of turning your head and eyes towards a visual stimulus. So if there's a flashing light behind you, you will turn your head and eyes to see what's behind you. Similarly, if there's a loud noise, you will turn your head towards that noise, and they are the inferior colliculi. So superior is to do with the light reflexes, inferior is to do with the auditory reflexes. So in other words, if you just it's very difficult to get selective damage just for such a tiny, you know, they are like pinpoints, they're so tiny. So by the time you damage the superior colliculi, more than likely you've already damaged the midbrain, and then basically I'm afraid that any damage to the midbrain is very, very severe and leads to coma. It may not lead to death, but unless the cardiopulmonary centers are affected in the medulla, but it certainly is difficult to just selectively damage the colliculi.
Okay, thank you.
All right.
We have Christy Tam. Christy Tam, you can go ahead with your question.
Hi. Yeah. So in the presentation, you mentioned something about neuroplasticity, and from what I know, correct me if I'm wrong, a big part of that plasticity is dendritic pruning. I was wondering if you could tell me a little bit about what the process of that pruning actually is like.
All right, so Rox, you want to talk about this? Should I take it?
I mean, I can have a go, and then if you know, you can complete me. So to cut a long story short, there's a continuous process of regeneration in the brain. Whilst neurons themselves cannot regenerate, at least not normally, different parts can. So you have a balance between enzymes that chew away and eat away at proteins. So you've got enzymes such as matrix metalloproteinases that just chew away at the building blocks, and you have enzymes that promote the synthesis, pruning, for example, of dendrites. And what you want is a balance where you get both these processes happening to allow some regeneration and pruning of these extensions. Does that answer it?
Yes, that does. Thank you.
Okay.
So we have another question coming up from Boy Canabasi. You can go ahead with the question. MK. Yeah.
I wanted to ask, you talked about us doing, you need to be able to do stuff continually, you need to do it repeatedly so that there'll be a lot of connections in the brain. Is it the same way it occurs when it comes to addiction? And why is it that it tends to be faster, that addiction for things, it tends to be faster in addiction than for normal body processes? I don't know if you can understand what I'm asking.
They're very different processes. So in terms of learned movements, you're relying a lot on, firstly, the ability to perform that movement. Yeah, so we've done the whole motor pathway. Secondly, for the cerebellum to then become almost like a hard drive that helps with getting better and better, faster and faster at that movement. Okay, that is very different to addiction. I'm not a psychiatrist, but you can get the different addictive substances in the brain. Okay, they excite certain neurons that then produce some neurotransmitters that make you feel one way or another, which is the effect of that drug. Now, the receptors for those drugs have a certain threshold, but if you keep hitting away at them, that threshold's going to get lower and lower. So, or in some drugs, it might get higher and higher. But addiction is the dependency of those neurons on getting the drug to then secrete a neurotransmitter. Nivedita, you're the neurologist. I can't go any further than this.
This is perfect. So in learned supervised training, you have the cerebellum that, you know, it teaches you and you become better and better at it. But for addiction, there are just completely different circuits that have little to do with the cerebellum, but with the basal ganglia and the frontal lobe. Remember the car accident and the frontal lobe damage? Those are the areas that are also important for reward and punishment. And that is a different circuitry with respect to learned information. So they're just two different pathways. Addiction will give you a high or whatever kind of pleasure it would give you. The other is completely different because it makes you better at performing something.
So we have a question from Jolene. Jolene, you can unmute yourself and go ahead with the question.
Yeah, first of all, thank you very much for the lectures. They were wonderful. So we've mostly been looking now at like the comparison of like a normal brain and then a brain with a tumor or Parkinson or Alzheimer. And I was wondering if there have been comparisons made between like a normal brain and someone with major depressive disorder, like if there is a difference in like active parts of the brain or something like that.
Jolene, sorry, can you repeat this question? Sorry.
Oh, no problem. But you can hear me, right?
Yes.
Yes, we can hear you.
Yeah. Okay. So I was just wondering if if someone has looked into if there are different if like how do I say it? Okay, so I was just wondering if someone has looked into if people with major depressive disorder, if in the brain some areas might be less active than they normally would be or more active than they normally would be.
Certainly more active, but not normally more active, aberrantly more active. I don't know if less active in maybe in depression. In depression, I think you do have less, yeah, because you have less serotonin, less secretion of the stimulant neurotransmitters.
So if I may add to this, Jolene, so in major depressive disorder, which is a disorder that a person is born with, okay? The two types of depression, you can be depressed because something happened to you, something major traumatic event happened to you, or a person could unfortunately be just born with the disease. When you are born with the disease, which plenty of the psychiatric diseases, for example, schizophrenia, bipolar disorder, etc., there's a problem with connectivity in the brain, and some of the brain areas just don't function properly. Okay, and again, I just want to also say that as we speak, these are things that we are learning as we go. We just don't have a clear understanding of all these diseases. However, we do know that there are certain areas of the brain that they just don't function properly. So, for example, there are parts of the brain stem that don't produce, as Professor Karare was talking about, certain stimulants like serotonin and dopamine, and so the patient just is depressed. You know, it just takes the things in negative way rather than being positive. There are areas of the brain that are just not perfused properly, that just don't receive a lot of oxygen, and so they can have more depression. You can actually work on neurotransmitters, and that is all what drugs are about. They will increase certain neurotransmitters, inhibit other neurotransmitters, and just for curiosity, so that we end up on a healthy note, if people do activities, and this has already been published, people do activities like meditation and yoga, they have actually the effect of reducing certain excitatory neurotransmitters like glutamate, and they actually increase inhibitory anti-anxiety, what was the term I'm looking for? Anxiolytic, right, anxiolytic neurotransmitters, they actually, help you sleep better, help you think properly. So that is to just complement the neuroplasticity and depression. You can actually modulate with neurotransmitters and get the brain to work properly as much as possible. But if there is an innate damage, it really becomes very difficult. I hope we answered your question, Jolene.
Yes, thank you very much.
I think we're going to have to wrap up now because we need to.
Sorry about that.
Sure.
To our
thank you, Professor Roxana.
Thank you again. Thank you again. And I'm sure we'll be able to stay in touch because you're all neuroanatomy enthusiasts. All right.
Thank you.
Bye-bye.
Bye. Bye-bye. Bye. Bye.